Our next session is imaging of the CNS and ME/CFS and will be moderated by Dr. Fred Friedberg. Fred?
Okay. I'd like to introduce Dr. Jared Younger, who obtained his PhD in experimental psychopathology at University of Tennessee in 2003. He then completed postdoctoral fellowships in pain medicine and neuroimaging at Arizona State University and Stanford University, before joining the faculty at Stanford in 2009. In 2014, he transferred to University of Alabama at Birmingham, where he currently directs the Neuroinflammation, Pain, and Fatigue Laboratory. His laboratory uses neuroimaging, immune monitoring, and clinical trial techniques to develop new diagnostic tests and treatments for pain and fatigue disorders. And today he'll be talking about neuroinflammation in ME/CFS. Jared?
Thanks. All right. What I want to do is present the data that suggests that ME/CFS involves brain inflammation or neuroinflammation, inflammation in the brain. I'm going to focus only on human neuroimaging. I'm going to talk about magnetic resonance spectroscopy, or MRS, and positron emission tomography, or PET, because those are the two modalities I think are best currently able to show us neuroinflammation in the living human brain. What I want to do is take you right up to where we're at today with those technologies in ME/CFS, and then I want to outline a few immediate next steps that we need to take. First thing I have to do is give a very, very condensed explanation of what I mean when I say neuroinflammation. I use that term pretty crudely just to refer to any time the microglia cells, which are the immune cells of the body or of the brain, move from that resting state, which you see in the top left, into this activated pro-inflammatory state, which you see on the bottom right. So obviously there's a structural change, but more importantly, there's a functional change. When they move into this state, they start to produce pro-inflammatory agents, like pro-inflammatory cytokines, IL-6 and TNF-alpha and interleukin-1 beta, many other things. Those cytokines, when they're secreted in the brain, change neuronal functioning in specific affective regions that cause what we call the cytokine-induced sickness response. That's heavy fatigue, kind of brain fog, confusion, low motivation, just general malaise. So if you get sick and you feel horrible, that's the product of these released pro-inflammatory agents in the brain. Now, these agents are supposed to be transient. So once that virus or bacteria is taken care of, the cell should pop back up into its resting state on the top left. What I believe is happening in an ME/CFS is the cells are stuck in that activated position, or they're flipping back and forth very rapidly. If that's true, we really need to know that, because if that's the case, there are strategies for targeting these cells that may be of benefit to ME/CFS. So we really got to see if that's the case. It's just a hypothesis right now. The problem is, how do we show that in a living human? That is very difficult. We don't have access to these cells. There's no ethical, practical way to access them in humans. So we have to come up with indirect ways to do this. And I'm going to show you the best attempts to do that. So one method that researchers are using is magnetic resonance spectroscopy. And this is a very cool extension of MRI. So you see MRI scanners all over the world. Most of them can do MRS as well. This gives you quantification of different metabolites, and many of them are related to neuroinflammation. So some of them are myoinositol. You see that more in glial cells than in neurons. So if you see a lot of myoinositol in a region, it suggests that there's glial proliferation or aggregation in that region. Choline should be, there's a certain level where it should be at. There's always cell turnover. This is kind of a marker of cell turnover. If that choline peak gets too high, it suggests that there's abnormally high cell turnover. That's typically a marker of neuroinflammation. And if it shoots up higher than that, it suggests there's a tumor, which is even faster. N-acetyl-aspartate is a marker of neuronal health. And so if you're in a gray matter region of the brain and that's going down, it suggests there's a neurodegenerative process. You're losing neurons. And then lactate should be very, very low in the brain at all times, because that really, you only see lactate if you run out of glucose. And so it's used as an alternative or backup energy source. So astrocytes and other cells only produce that when the metabolic demand exceeds the glucose supply. And really, there's only a few reasons why that would occur in the brain. So there's been several studies, actually, to look at these chemicals in the brain and ME/CFS. The most recent one outside of our group, Nadelson and his group did this one, where they looked at lactate in the cerebral ventricles. And these are kind of the cavities of the brain where the cerebral spinal fluid circulates. So they reported that there was higher lactate in the ventricles in chronic fatigue syndrome and in fibromyalgia and in people with both. And it's about 3 times higher than in healthy controls. That's pretty significant. I can only think of about 4 reasons why you would see that. One would be if you're hypoglycemic, and that's easy to test. We can rule that out. The other one would be if you have a vascular problem, and so you're not getting enough blood supply to the brain, and we can rule that out too with a cerebral perfusion scan. The other one would be if you have a tumor, but then we can see that with structural scans. And so once you rule those things out, what's left is neuroinflammation. So I think this is really strong evidence of that. That's not the first time it's been reported, and I don't know if Michael Van Elzacker is going to dislike me for this, but I'm taking a table from his recent paper that he's probably going to refer to. So there's one table I just had to take. It just came out the last 2 months, a very nice review. And what I really like for my talk is the fact that it identifies that lactate is probably the most commonly found disrupted metabolite in ME/CFS. And I think that's the strongest evidence to date that there is something wrong in the brain of ME/CFS, and it involves neuroinflammation. You can see this has been found in addition to the ventricles. Groups have found it in the occipital cortex. They found it in the anterior cingulate cortex, which is really important for sickness response. And they found it in the corpus callosum. So this has been found by many groups. What we wanted to do is take that study, and instead of looking at discrete regions of the brain, which is the classic way of doing MRS, we wanted to look at the entire brain. And so there are some MRS techniques that allow us to look at the whole brain in 3D with about 3 to 4 millimeter voxels. And so now we can get these maps. Instead of looking at just the cerebellum, let's see where the elevations are. So we're doing this with an echoplanar spin imaging technique. This was developed by Andrew Maudsley at the University of Miami, and it's a very cool technique. Uses a lot of kind of acceleration factors and shortcuts to get you a lot of data in a short period of time. So very nice MRS technique. We have a paper based on this. I hope some of you got to see Christina Mueller's talk, who's our senior grad student who ran the study. She did a presentation. There was also a poster. If you missed those and you want more details, the paper is out and you can look at that. Let me show you just a little bit, because I do want to talk about PET stuff. So I'm going to give you a very condensed overview of what we found. The very short story is we found lactate all throughout the brain in ME/CFS. What you're looking at here is the average of the ME/CFS patients, 15 of them on the right, and then 15 healthy controls, age match, gender match. These are all women on the left. And on the far left, you see the regions where there was a statistically significant increase of lactate. Lactate was elevated throughout the brain. These are the regions where it was statistically significant, but it was pretty much throughout the whole brain. Many areas, I don't have time to go into the importance of these regions, but things like the anterior cingulate cortex, you see the thalamus, you see the hippocampus. These are very key regions involved in the sickness response. And so it's very exciting to see those. I will note that on the healthy group, you see that little ring of red on the outside. We're still working out some problems when you get close to the skull. Those are erroneous high values, and we have ways to throw those out. But there's still a few things to handle kind of when you're close to the skull. I do not think that's legitimate lactate, but all the stuff internal in the ME/CFS looks legitimate. And those lactate values are about 4 times higher in ME/CFS than controls, which is what the previous groups have found. Very, very consistent. The next thing that comes out in the review is choline. And that's been reported by 3 other groups. So we looked at choline, and we also found it was elevated in several brain regions. Anterior cingulate cortex, middle cingulate, again, key sickness response regions. Looking at ME/CFS on the right and healthy controls on the left. And those red areas are where the choline peak gets as high as the creatine peak, and that tells you something abnormal is happening. It really should be more like 0.5. It should be about half. So this is suggesting in these regions, for some reason, there's very high cell turnover, and that suggests a neuroinflammatory process. The focus of this was the anterior cingulate cortex, and in particular, the very bottom part of that kind of white region of interest is the subgenual ACC. And this is a very important sickness response region. That's where kind of depressed mood and malaise is centered. And so if this comes up in a lot of different regions, but a lot of the affective experience of the sickness response will involve that region. And you can kind of see what a healthy control looks like. You can see that choline peak is about halfway up the creatine peak CR, whereas with the CFS patient, they're kind of matching. I don't have a laser pointer, so just follow my finger. And then on the right, you can see the distribution of control in ME/CFS in that one region. They are not separating completely in any particular region. So we don't have 100% separation, but there is quite a bit of separation. So just wanted to show you what the distribution looks like. We also get temperature. And the reason we wanted to look at temperature, it's never been looked before, is because we believe if there's this excessive metabolic demand, that may exceed the cooling blood's ability to reduce that heat. We didn't know if that would turn out to be the case, but we're able to get whole brain temperature with the same scan, and we found that the brain of ME/CFS is running hotter by about 1 degree Fahrenheit. There's many areas where that increase is significant from healthy controls. You can kind of see this in ME/CFS on the right. The higher temperatures are in red. In particular, the insula and the putamen and the thalamus, those are the same regions that had increased lactate. And so we're getting convergence where there's one side of neuroinflammation, there's this other one as well. So what needs to be done next? There's 4 things we need to do with these scans. We need to do it in a larger sample size. 15 versus 15 does not tell us what's happening in the population. An important part of that is we need to look at subgroups. Clearly, not everybody with ME/CFS is going to have this increased lactate or increased temperature. We need to know the importance of that. Does that mean some people with ME/CFS have neuroinflammation and others don't? Are there different types? We have to figure that out. We also need to test it longitudinally. We do other tests where we look in the blood and we test people over many months, and we look at their good days versus bad days to see what tracks with those good and bad days. I believe this brain scan will show us what we should see on a bad fatigue day. We should see elevations of these metabolites, these neuroinflammatory markers, and they should be suppressed on a good day. If we see that, that's pretty powerful evidence that what we're seeing in the brain is strongly related to the symptoms. Last thing is we need to validate the scan with a PET gold standard, and that takes me over to the PET stuff. So positron emission tomography, if you're not familiar with it, very cool technique using radioisotopes that you can link to different ligands to go to different receptors. And there's many other things you can do with that, but that's one of the ways that we're using it. So the really nice thing about PET is, remember I said we can't look at the cells directly as they change shape, but fortunately when those microglia move into that activated position, they overexpress a receptor called the translocator protein receptor, TSPO. We think those are to provide breaks so other cells can push that microglia cell back to the resting state or to an anti-inflammatory state. But regardless of the function, when the microglia are activated, there's more of these TSPO receptors. So if we use a radioisotope connected to a TSPO ligand, we can inject that, and if we see a lot of uptake of that ligand in the brain, that suggests more microglia are in the activated position. So this gives us a marker of activated microglia. This has been used in a lot of different groups. This is what happens to National Football League players when they've had their head banged around for many years. When they're scanned 3 decades later using this technique, there's evidence. You see the NFL players on the right. This is an average of, I think it's 30 versus 30 healthy controls. I could be mistaken, but a pretty good sample size. You see increased uptake of this marker, which suggests activated microglia, as opposed to the healthy controls. That suggests that that brain damage gave persistent neuroinflammation through the rest of their life. It may explain why they have increased incidence of mood disorders and cognitive disorders. This has also been done in ME/CFS. Most of you have probably seen this study. This was a 2014 study by Nakatomi and colleagues, and it's the only PET study to look at neuroinflammation in ME/CFS. They found several regions: midbrain, pons, thalamus, cingulate. The amygdala is in there, but to my eye, it doesn't meet statistical significance at P05 if you care about that. And then the hippocampus. So at least 5 regions where the ME/CFS patients had an uptake of this microglia marker. What's really nice about it is it overlaps almost perfectly with what we found with lactates. Thalamus, cingulate, hippocampus, same places we found upcrease of lactate. Now the midbrain and pons we have not looked at specifically, and so we need to do that. We may find lactate in those regions as well, but there's some special techniques we need to do to look in those regions. And on the right you can just see that thalamus in yellow, and you can see that they had perfect separation between groups. Now, of course, as you know, if you run a test and you get 100%, as you just heard, separation between groups, you've got something really, really important. So this study has to be replicated. And the reason it needs to be replicated is because they used a very old marker for TSPOs called PK11195. And while it was great at the time, it's non-specific. It doesn't give us a high degree of confidence that what we're seeing is activated microglia. It just docks on too many things. So now there's second generation TSPO markers that we have access to that are much more specific. So we need to replicate that study and see if it holds up. If it does, we'll have some very strong evidence that ME/CFS involves neuroinflammation. Now, we received some pretty good news on R01 score, which involves this PET imaging. So I hope soon we'll be able to replicate this and hopefully show pretty definitively if these individuals have neuroinflammation by activated microglia. And we plan on doing these scans on the same people who are getting the MRS scans. And so we can see, are these different modalities providing complementary information. It's going to be really, really interesting to see. I also, as you heard, I think yesterday, Jose Montoya is using a similar radioligand, whereas we're going to look at moderately severe and severe patients, so people who can get to the lab by some means. Jose's group is going to look at homebound and bedbound. And so since we're using pretty similar techniques, that's going to provide some really interesting information about, are the very, very severely ill suffering from the same thing as the people who can make it to the lab? Is it just a matter of severity of the illness, or are they different things? And I think converging this evidence or this information is going to tell us that. A couple more things about the PET. We know that these TSPO ligands are showing us when microglia activate. And this group, this is San Diego and colleagues in 2015. I really love these kind of studies. They injected people with lipopolysaccharide or endotoxin. And if you don't know, that's kind of the part of bacteria that our immune cells use to recognize when we have an infection. So if you inject people with lipopolysaccharide, you get a very robust immune response peripherally, and you also get the microglia response. So they showed that when you inject people with lipopolysaccharide and you do one of these TSPO scans, you see very rapid increase in the microglia activity. So they seem to be very quickly moving into the activated position, which is exactly what you would expect because people report feeling really, really sick, as they should. So the microglia are activating, that's that bottom row right there, producing those inflammatory signals, and then the people are feeling really bad. What's missing here is no one has ever done this in fibromyalgia, and they've never done it in chronic fatigue syndrome. And what I think would happen is if we did this in healthy controls versus ME/CFS, I think what would happen is the ME/CFS group would show an even exaggerated microglia response to that LPS injection. That would be really cool to do, and I think it would distinguish the groups, and that would suggest the microglia are primed or hypersensitized, which is one of our hypotheses about what's going on. They react to little triggers in an overly robust way, probably daily triggers that normally would not trigger an immune response in us, so the person constantly feels sick when they don't actually have a pathogen. That's one model. Just something I want to note really quick before I do a few wrap-up slides. We do inject people with lipopolysaccharide. We have not combined it with imaging. I've just finished a couple of weeks ago this in fibromyalgia, and I just want to throw this out here because it is relevant. When we inject people with fibromyalgia with about 0.04 nanograms per kilogram of lipopolysaccharide, we see that they have a suppressed kind of typical immune response. So there's less TNF alpha, there's less IFN gamma, there's less interleukin 1 beta. All those typical inflammatory things are actually suppressed in the fibromyalgia group. But they have a huge, weird alternative inflammatory reaction that is mostly marked by a massive increase of leptin. And you've heard leptin before, and it's because everywhere we look in ME/CFS and fibromyalgia, we are finding leptin. If I track people over time, the best predictor of their day-to-day severity of fatigue is leptin. You've heard Jose find leptin cross-sectionally. Now we're finding, by the way, what you're looking at, this is baseline-centered leptin. The green line is fibromyalgia. The blue is healthy controls. It's at baseline, and then we injected them with LPS between time point 1 or 2, and these are hours. And so you see this immediate increase of leptin, and then you see the second kind of increase that comes later. Now we know this isn't circadian rhythm, and we know this isn't because we gave them a little meal at time point 5, because we also injected them with really, really low endotoxin, and this response does not occur. The groups are the same. So this is due to that kind of low-medium level of LPS. The reason I mention this is because I think this ties in really well with the central imaging that we're doing. Again, if we can combine these kind of immune perturbation studies with the imaging, I think we can look at the abnormal microglia response. So one model from this is that the peripheral immune response is muted, but a microglia-mediated response is exaggerated. So they would look less sick peripherally, but they would feel really, really sick because of that microglia response. So that's what we're working on. Now this was FM, not ME/CFS, but we need to do it in ME/CFS. Some of these people meet the case criteria, but most of them didn't, so it needs to be replicated. I don't want to assume they're the same disease. I think they have a lot of overlap. But last PET study, and then I will do the wrap-up stuff. We just got funding from ME Research UK to do a different PET study that's really cool. What we're going to do is draw blood, isolate the leukocytes, T cells, B cells, things like that. We're going to incubate them with ZR89 oxine, which is a very long-lasting PET tracer. So that goes inside the cells, then we re-inject those cells in the patients, and we wait about 5 days and then scan the brain. What we're looking to see if any of those peripheral T cells and B cells breach the blood-brain barrier and pop up in the brain. If we see that, that's going to be really compelling evidence that there is a pathological, serious pathological immune-related process occurring in these individuals. What I think we'll see is the healthy controls will show absolutely no signal because those cells are not infiltrating. And in ME/CFS, we will see some sort of signal. I don't know if it will be small, medium, large, no idea. We'll find out. We will probably start this in 4 months, just working out a few more things, and then we'll get this going and see what we find. Here's my model to bring this all together, what I think is happening. I believe, just based on the symptomatology, I believe that neuroinflammation is the most proximal cause of the symptoms of ME/CFS. The fatigue and the cognitive issues, everything that most of the patients report, I think neuroinflammation is one step from that, and the cytokines that are released. However, I think there's multiple reasons why someone would have neuroinflammation, because these microglia are supposed to be responsive to a huge range. In fact, they're supposed to be responsive to everything that could damage your brain. And so it makes sense that there's many reasons why they might be sensitized or activated. So just as an example, here's 3 potential pathways leading to neuroinflammation, which would lead to symptoms. And so what I think is happening is there's a core set of symptoms that all ME/CFS patients have that are driven by the neuroinflammation. And then there's these kind of different symptoms that some people have and some don't. And I think those are driven by whatever is pushing the neuroinflammation. So just a few examples. That could be a pathogen, viral, bacterial. If you have a recalcitrant, resident pathogen, that will easily lead to this microglia activation. It could be an autoimmune disorder like we'd see in rheumatoid arthritis or multiple sclerosis. It could be autoinflammatory and involve abnormalities in mast cells and complement proteins. It could be something, an inflammatory gut problem or some other peripheral inflammatory thing that's causing the central nervous system to release cytokines as well through the vagus nerve or through some other pathways we know of. It could be these dysregulated fundamental systems we keep talking about, mitochondrial problems, energy transfer problems that would cause the immune system to behave abnormally. It could be a problem with the vasculature, with the blood-brain barrier, something like that. There's probably, I could fill up slide after slide of possible causes. I think what I want to say with this is, I believe if we find this, if we can treat the neuroinflammation, I think we will alleviate a lot of the symptoms in the ME/CFS patients. In some people, that will probably be sufficient to cause a remission of the disease. In other people, there will still be a fundamental issue that needs to be fixed, even if we alleviate some of the symptoms. So we don't want to have a condition where, if you've got your smoke alarm going off at 3 o'clock in the morning, because your kitchen's on fire, you don't solve it by taking the batteries out of your fire alarm. You still have a problem you have to fix, right? And so if there's this fundamental problem, it has to be addressed as well. But I think in any case, calming down that neuroinflammation is going to help people feel better. So I think it's something we need to tackle. Last slide, we're going to be hitting these studies really hard over the next few years. My goal, I like to set public goals because it keeps me anxious about meeting them, is in 3 years, I want to know definitively if ME/CFS as a group involves neuroinflammation on the whole. And then in 5 years, I want to have a definitive, reliable, valid neuroinflammation scan that can work on the individual level. So we can take as many people as you want, put them in the scanner and say, this person definitely has high, medium, or low neuroinflammation or not. That's the goal in 5 years. And now I think we have the resources to do that. We just got to really put in the time every day to do that. We will move as fast as we can because, again, there are treatments both existing and things that can be developed that we can use to target this if we're sure that's the thing that we need to target. So if anyone has expertise in advanced neuroimaging or in helping us interpret some of this inflammatory stuff we're trying to work out, feel free to contact me. I would love to collaborate with people that can help me think through and get to these advances even faster. So that is what we're working on.